In this module, we will discuss the mechanism of meiotic recombination. We have been discussing the homologous recombination, different its functions, how it is performed, and now we will study the mechanism. What are the mechanism involved in the meiotic recombination, mitotic combination, and what mitotic recombination is? As a matter of fact, mitotic recombination is a type of genetic recombination. This process shares many characteristics in common with homologous recombination, which we have discussed in other modules. As seen here, there is a homologous pair of chromosomes, and there is homology, which is considered in the sister chromatid. They further overlap with each other, and eventually a new chromosomes or modified chromosomes, they are produced after the recombination, which is at the bottom of this picture. Now, during meiosis, the synapsis is term which we have studied earlier. That is the pairing of homologous chromosome. When chromosomes they pair together, that is termed as a synapsis, and it ordinarily proceeds the genetic recombination. This is nothing but a genetically recombine of the chromosomes together. Now, the mechanism involves that this study, meiotic recombination, was extensively studied in the budding yeasts that are Saccharomyces cerevisiae. The process starts with a chromosomal lesion. On the chromosome, a lesion occurs and a double-stranded break of the DNA present on the chromosomes, it is initiated. Next, an exonuclease enzyme, it recognizes the break. Now, these enzymes recognizes the break at the specific site at the 5' ends of the two of the strand. Remember, if we have the two double-stranded DNA, the nick occurs, it breaks, then we will end up 2 5' ends of there. Now, this nucleases, it recognizes those, and what it does, it takes them, and the two of the strands, it creates them into the 3' single-stranded overchange. ये उनको 5' से 3' में change कर देती है nuclease. Just as shown in this case, you can see that the double-stranded break occurs on the red, then that break on the bottom, there are 3', 5', blue and red, and further it produces a D loop where, and it joins them together, and a new gene or a new double-stranded DNA is produced, which is embedded in the gene. Now, one of these single-stranded ends, they can then invade the other DNA duplex. जो single-stranded बन गया, वो क्या करता है? वो दूसरे DNA duplex जो है, उसको invade करता है, उसको जोड़ता है, forming a D loop as we observed in the bacterial homologous recombination, that the displacement loop, D loop means displacement loop, it is formed and it is recombination over there. Just as this is the model for the meiotic recombination in the yeast. And as you can see, a DNA repair synthesis over the top, it is there. Then in the red, a D loop is formed. Further, it is going over there, and it invades the strands, making the crossover recombinations, which are left and right. That is the resolution crossover A and B occurs, and new strands are produced over there. The classical work on meiotic recombination, क्या हुआ, was performed in the budding yeast, as I've already indicated, and the double holiday junction to be predominant event. Remember, the double holiday junction, which is formed during this process, is a predominant event during this recombination. Now, as it is shown in this diagram, there is a double holiday junction which is being produced on the top, and then further 4 new recombination of the molecule is being formed. Additional function of homologous recombination in eukaryotes is that it has additional function in eukaryotes. It produces additional function. उसके अलावा जो है वो function पैदा हो जाते हैं in the eukaryotes like yeast, and it is required to pair homologous chromosome in preparation for the first nuclear division after segregation during the meiosis. This is the mechanism where before the nuclear division occurs, what it does, it pairs the homologous chromosomes in preparation for the first nuclear division. Remember, there is a division in the meiosis, but before that meiosis occurs, the homologous chromosomes, they are paired by the homologous recombination mechanism in the cells, just as seen here. On the left, there is a diploid cells with pair of homologous chromosomes, as seen. It is joined together, it occurs, the mechanism occurs, and the site of the gene conversion is there, site of the gene conversion, left, right, new genes, they are produced, and the meiosis produces the haploid cells with chromosomes that have crossed over on there. So from 1, there are 4 haploid chromosomes, they are being formed, and they will be under the meiosis. Meiotic recombination also frequently give rise to crossing over. जाहिर है कि जब क्रॉसिंग ओवर होगी तो फिर जीन जो हैं वो होमोलॉगस पेरेंट के ऊपर मूव होंगे. So meiotic recombination also frequently give rise to crossing over between genes on the 2 homologous parental chromosomes. Homologous जो parental chromosomes होंगे उनके ऊपर वो crossover होंगे. And homologous recombination has additional function in eukaryotes. It is required to pair homologous chromosomes, जिस तरह पहले बता चुका हूँ, in the preparation for the first nuclear division of the segregation of the meiosis. Meiotic recombination, चुनाचे, also frequently give rise to crossing over between genes on the 2 homologous parental chromosomes. So this is the basic end result of this meiotic recombination, that it frequently give rise to the crossing over between genes on the 2 homologous parental chromosome before the meiotic division occurs.